Welcome to channel Toxicology on Board. Today in this video we'll be learning about DABT question DNA microarray. Let's directly go to the question. Let's see what is the question. In risk assessment, what results can be obtained by using DNA microarrays in toxicology study? So in case of ABT exam, no, risk assessment is one of the very important topic. In that also they are coming in case of risk assessment, no, instead of going for traditional methods, new methods are also being implemented for the risk assessment. See, earlier the risk assessment was based on Lowell approach. Okay, now we have BMD approach is also there. Now we have TD50, that approach is also there. Now to apply for risk assessment. Now in like that only the DNA microarray or whatever your high throughput screening methods are there, no, which were initially used for drug discovery. Now they are also implementing the same kind of tools for your risk assessment also. So how that is, how they are framing a question around this concept will be learning. So in risk assessment, what results can be obtained by using DNA microarray in toxicology studies? Okay. So we have option A, many genes can be evaluated simultaneously to detect the patterns of response to a toxicant. So how a toxicant is responding to the test system, you will understand the patterns by using the simultaneously you can read many genes because thousands of genes will be there. So all those things can be done in a single DNA microarray assay, and you will be getting patterns of this response you will be getting from this kind of assays. So what is the second option we will see? Detection of differential gene expression, eliminating the need to test the toxicant in the whole animals. My understanding is that complete elimination of the animal studies will not happen. That is the reason probably this answer, second option might not be applicable to the this particular question. Next, we have changes in the gene expression can be precisely correlated to changes in organ function. I think here again is talking about target organ as also is talking about precisely. For that reason, this is the option also not applicable for this question. Last option is measurement of gene expressions that can be precisely quantified following exposure to a toxicant. Now, based on new type of tools and data interpretation, we are able to quantify the results from this DNA microarray also. See, for example, you are doing a traditional study, you will get a Noel, you will get a Lowell, you will having a dose with exposure with response you can do, you will getting a number, quantification will be there. If you do if you do a genotoxic assay like Ames assay, chromosome aberration assay, or other genotoxicity or mutagenicity assays are there, no, out of which you will get either results in negative or positive, but you will not get the dose. Of course, there you will be concentration will be there. 2000 microgram per plate, 5000 microgram per plate will be there. But that dose, for example, the genotoxic dose cannot be used for risk assessment. But again, I will say there are some articles and publication where NOGEL value is also available. So any statement which I am saying, there is an exception to that. So probably this last option might not be applicable for this question because precisely quantified following exposure to a toxicant. For that reason might be there. So let's see what is the answer. So what we are doing, the question is risk assessment. What results can be obtained by DNA microarray in toxicology studies? So the right answer, as you can see in the green text, many genes can be evaluated simultaneously to detect patterns of response to a toxicant. Here the toxicant might be any chemical or any pharmaceutical pesticide, anything, right? What is the explanation reference given by ABT? This is the ABT where 50 questions are there. A series of questions was available in the DABT site. Sophisticated analysis of microarray can be used to analyze patterns, gene expression between comparative sets of samples. Okay, there are different sets of samples will be there, right? These analysis can be linked to functional interpretation and classical toxicology endpoints. What are the classical toxicology endpoints? That is nothing but LD50, MTD, NOEL. These are the classical toxicology endpoints. These functional interpretation can be correlated with the classical toxicological endpoint. The results obtaining from DNA microarray, how that results can be useful for risk assessment, that is a concept what we are trying to understand. Okay, don't get confused. So let's see some more things that we can learn around this concept. We'll see. Okay, micro, what is for certain students who are not at all aware of microarray, let's go briefly what is microarray. Microarray analysis for risk assessment require analysis beyond the cluster analysis. To do normal type of drug screening analysis with a cluster in a group of chemical group, this NHO group or SS thiol group, sulfide group, like that. Normal basic cluster of analysis is not just enough for microanalysis. Okay, requires analysis beyond the basic cluster analysis to arrive at a functional interpretation and linkage to a conventional toxicology endpoints. Conventional, I already told you, LD50, MTD, NOEL, LOEL, right? Next, we'll see because of vast number of measured responses with gene expression arrays, pattern analysis technique. In the option also we see pattern analysis techniques are being used through pathway mapping platforms such as MapFinder and GenMap. So go back and read in the in at least have some idea what is MapFinder, what is GenMap. These are different platforms, mapping platforms, gene mapping platforms, as well as big data approaches. Now, what is that? We have thousands of genes are there. Their response are there. We are able to make cluster analysis. We are able to make a mapping, a decision tree like this. We are able to make. So all those things can be learned in this particular map finder or GenMap. The Gene Ontology Consortium has developed a controlled vocabulary ontology for sharing biological information across species, across mice, rat, rabbit, how that particular drug is affecting in the mice, in the rabbit, like the different across different species. Okay, because there is a little, we are all same. All species are all same. At the same time, we are also not same. Okay, basic pattern might be same, but there is a genetic variation will be there with among the species. Okay, database users can then annotate um, their gene products with gene ontology terms, okay, which is given in this one vocabulary ontology, establishing a consistent description and definition across database and studies, and this facilitates the risk assessment. How the gene, we are now able to understand again how it is applied to the risk assessment. Next, we'll see through the power of this gene ontology terms, analysis methods are being developed to allow for quantitative time and dose dependent interpretation, right, of genomic response using a system based approaches to integrate response at the biological and cellular levels. Okay, both at biological levels as well as at the cell level. Okay, an example of integrated analysis of multiple omics data set is concepted. Please go back and read about this one. Don't get panic. All these new might be some terms for some people. Those who are working in genetic area, those who are working drug discovery or screening area, they might be aware of these terms: proteomics, genomics, metabolomics. For some people, it might be something little bit of new. So need not worry. You can pause the video, go back and read in detail. And this is the 2017 NRC report provides a new framework for incorporating this information, whatever the results which we are getting from DNA microarray, such kind of assays across various levels of biological observation. There will be uh, there is in this particular 2017 NRC report, you can see like the reverse will be there. I'm not getting omics application of omics will be there, will be discussed in detail here. Okay. Then next we'll understand one in little bit more detail about this microarray. A microarray is a laboratory tool used to detect the expression of thousands of genes simultaneously. As I said, no, then next, it consists of a solid surface like a glass slide will be there. Okay, with thousands of small spots will be there like this here. Okay, each containing a known DNA sequence. Okay, because of the advancement in genetic technology, we are able to identify it. Now this slide. It is already containing the spots, and on which each containing a specific DNA sequence probe is already there on this particular mounted slide. Okay, by hybridizing labeled DNA RNA samples to the probe. Now we are taking our samples which is containing the DNA or viral sample or the RNA sample to the probe. Researchers can measure the relative abundance of different genes in a sample, right? Then next we'll see high throughput. This terminology, this term you might have come across during your drug discovery when you will be reading about drug discovery, high throughput screening where attrition of molecules will be there. Out of thousands of molecules, only few molecules, only up to 10 1 to 10 molecules will only reach the final stages of drug synthesis. So microarrays can analyze thousands of genes at once, making them suitable for studying complex biological systems. Okay. Parallel analysis, all the probes on the microarray are analyzed simultaneously, allowing for a comprehensive view of gene expression. So together all this data you can see on a single this thing. So comprehensive view of the gene expression can be seen. Parallel analysis, how that is affecting liver, how it is kidney, all these things you can do parallelly, you can observe. Parallel analysis meaning here it is. Quantification, the amount of hybridization between the labeled sample and the probe on the microarray can be quantified, providing information about the gene expression level, right? So hope from this video you will able to understand about DNA microarray, how there are new concepts are coming like genomics, proteomics, metabolomics. These are the new type of science are there which are now having a key role in the case of risk assessment. You have to understand. Now we are in the era where we are said not to conduct toxicology studies in animals because of the ethical reasons. Okay, so we have to implement 3R 4R principles: reduce, refine, recycle, rehabilitation. There are some 3R principles are there. Okay, now if we do not conduct any toxicology studies, then we need to have some data in our hand to analyze, to apply it to risk assessment. So how these type of new type of sciences are there? Organ on chip technology is there. This metabolic proteomics, some kind of data you will be getting. Nowadays you can use the software and predict the LD50. You can predict the NOAL also. So those who are people working on ECA rich dossier, they will be understanding that a lot of softwares are available where you can get the NOELs also, NOELs. And as I was telling in my first slide, no, earlier genotoxic mutagenicity, there was no dose related response for the concentration response was there, but you will not have a typical dose response curve with respect to gene tox or AIMS assay. Like this 50 mg dose, like this, there was no results. But now I have come across certain papers where I think that is example for ethane methane sulfurate EMS. I was trying to calculate permitted daily exposure for EMS, where I have there seen some kind of toxicologists who have done risk assessment considering NOGEL, no observed genotoxic effect level. Okay, NOGEL. Earlier, we never used to come across this kind of term because we were only coming across NOAL, NOEL, LOAL like that. There was no NOGEL. But now we are coming so even genotoxic studies also. So similarly, this coming results coming from microarray, they can also be quantified. They can also be have a key role in the case of risk assessment. Okay. So new types of technologies, new types of alternative methods, how new types of new science can be used, and its role in toxicology is very, very important. So from that area also you'll be getting one question. Okay. Thank you so much. Be curious. Please write mail to arya.priyanka18@gmail.com for any queries, and also like, subscribe, and follow my YouTube channel, Toxicology On Board. Thank you so much. All the best for exam.